Okay, we're going to try something here. Last week we had a couple of snow days, so we're getting a little bit further behind. And to save on some time, I thought I would throw this together for you guys to watch at home. It goes into that Nufke presentation that we started before we went to the computer lab, but we sort of took a pause while we went and did the Howard Hughes activity. So we're going to finish it up right now, and then I have a couple animations. Again, this is actually on the website, so if you do not remember the first part, or you were gone that day, or you just need to go back through, please do. We started talking about the cell and the cycle, the reasons why a cell would divide. For growth, we have for growth or for repair. We talked about internal and external controls and regulations, especially the MPF, that's the cyclin and CDK complex. There is a good chance that a question about the relationship between CDK and cyclin will be present. It is a good relationship to know and understanding this graph, and I'll have an activity for you to sort of review it. We have the external controls like the platelets, derived growth factor, and we talked about density dependency and anchored mechanisms, factors that control it. And then we stopped here at cancer. We went to the computer lab. We had this little click and learn activity, if you remember, to learn about the eukaryotic cell and cancer specifically. What you should have gotten out of this, though, is that mutations happen. Mutations are responsible for the triggers when it comes to cancer and the regulation of cell cycle. Mutations happen all the time. Mutations may be positive, they may be negative. It's just going to happen. We're going to talk more about mutations. We talked a little bit when we looked at evolution. We're going to talk more about mutations in a few weeks. And I'm going to actually pause this for a second.
Mom, can I?
Honey, I need you to go out.
But can I do ABC Monster?
Okay, so I do not know if this is going to be changed up or not. We'll see. I'm sorry, I had to pause it for a second. Okay, so let's get back to this. Two types, if you see a little head in the background, it's one of my daughters, sorry. Two types of genes that factor into cancer that you guys looked at. We had the proto-oncogenes, which is the gene similar to putting your foot on the accelerator. They speed up cell division. It makes it go to the next level, puts it in hyperdrive. In oncogenes, the mutated versions, they're always on. And you only need one of the alleles to be mutated for this to be on. Or for this to, when I say on, it means that it's going to increase it. Since you only need one allele mutated, it is a dominant form. Now, the other end of the spectrum, I'm going to skip through this for right now. We're going to come back to this when we talk about molecular biology, so don't freak out when you see translocation, gene amplification. You may remember some of this from genetics or regular biology. But I want to show you the other one that you saw in the HHMI activity, the tumor suppressor gene. This is the analogy of putting the foot on the brake, putting your foot on the brakes of the cell cycle. Normally, it stops. These genes stop the cell cycle from proceeding to the next checkpoint or proceeding through the next checkpoint. However, this is a gene that is recessive. Both alleles need to be mutated for it to be stopped. But once both mutations happen, it completely stops or it's turned off, I should say. It's turned off, meaning that it allows the cell cycle to go on. Normally, it stops the cell cycle from proceeding. Two mutated alleles, though, and now all of a sudden, the gene is not functioning, and the cell cycle is able to proceed on. Okay, we'll come back, as I said, to this. P53, I hope that does ring a bell. P53 was one of the regulatory processes that was in this chart that you filled in. Okay. There is a nice video if you want to look at Gleevec. It is a cancer treatment, and you do want to have some ideas of cancer treatments. They have a couple in the book, but there's a good video on one. Big ideas that you can take away from this is you need to know the mechanics of the cell cycle and the checkpoints. You need to know the MPF, the cyclin, the CDK complexes. You need to be able to understand proto-oncogenes and tumor suppressor genes and the role that they play, and have a kind of familiarity with cancer treatments. Now, hopefully this works. What I'm going to do is show you a little video on, oh goodness, let's see if it works.
The cell cycle is controlled by an interaction between cyclins and cyclin-dependent kinases, CDKs. During G1, the cell gradually accumulates cyclin, which binds to CDK to form a complex. When the level of the cyclin-CDK complex exceeds the threshold, the G1 phase ends and DNA replication begins. Cell division is normally turned off in healthy cells by proteins that prevent cyclins from binding to cyclin-dependent protein kinases, CDKs. These proteins are called tumor suppressor proteins. Among the most widely studied tumor suppressor proteins are the retinoblastoma, RB, and P53. The unphosphorylated RB protein binds to transcription factor E2F, a protein which activates genes in the nucleus.
So RB is associated with G1 cycle.
The G1 checkpoint is passed when CDK interacts with cyclins to phosphorylate RB, releasing E2F. The protein P16 reinforces the tumor-suppressing role of the RB protein, preventing its.
You don't need to memorize all these P16, E2F. If you have an idea of RB, and then P53 up over here, P53 shows up in multiple checkpoints.
The integrity of the DNA and activates the DNA repair system if the DNA is damaged. P53 stops the cell cycle at the G1 checkpoint to allow time for the DNA repair by inducing the transcription of P21, which binds to cyclins and CDK and prevents them from interacting. Without the CDK cyclin complex, the cell cycle cannot proceed to the S phase, and DNA replication cannot occur.
Okay, P53 also comes into play at the G2 phase. Before it goes into mitosis. It's another checkpoint for DNA repair right here. So there's actually a couple of checkpoints for that. The very first one, RB, again, was the very first G1 checkpoint. And then one other thing, and I'm going to stop it here, is there is a game for those of you that like to play a little bit. It's called, if you go to nobelprize.org, control of the cell cycle. Now on my Mac here, I do not have the plug-in, so I'm not going to be able to show it to you. However, it's a fun little game if you're looking for a way to decompress. And also, check how you're doing with your steps of the cell cycle. You actually have to regulate it. And actually, nobelprize.org has a lot of different games and a lot of different information if you search around on here that you can find out. And I hope this helps you. I'll post it on the website. Again, Nessa's Prezzies are on the website too, okay? And I will answer any questions that you have in the next class. Thanks.
Are you done?
Just a minute. Okay, it's going to take a second. I think I have too many going on right now.